And our next speaker is Professor Terry Jernigan, and she's a professor in the cognitive science department, but she's also in psychiatry and radiology, and are there any other departments I've forgotten about? And she's the she's also the director of the Human Development Program. And she's going to be talking today about some of the stuff that they've been working on. If we get our technical side in order.
You got it.
Well, looking good. Okay, so I give you Professor Jernigan.
Thank you. Well, I knew these guys were going to be a hard act to follow, and they are. But it's a privilege to be able to represent cognitive science today, and a pleasure for me to describe for you a program of research that my colleagues and I have been developing in the Center for Human Development, which is one of UCSD's interdisciplinary organized research units. And I'm going to give this talk a little bit at 30,000 feet, but I hope that if nothing else, you'll take away from it a new model that we're trying to adopt that we hope to use to create a more integrative, high-dimensional research paradigm for the study of the developing human mind and brain. And I'm going to start by telling you a little bit about the conceptual model that guides our research program and about some of the questions that we find most highly motivating in the CHD. So we think of ourselves as studying behavioral phenotypes, and a kind of slightly less wonky term for the focus of our research is individuality, really, by which I mean relatively persistent behavioral attributes that distinguish otherwise demographically similar people, such as things like emotional or social or cognitive biases, or things like the accumulated domain-specific knowledge or expertise. Can be things like literacy or mathematics knowledge, or the level of fluency of mental skills like calculation, things like that. But it's important, we think it's extremely important to bear in mind that these kinds of behavioral phenotypes are really only meaningful within a developmental context. And so obviously proficiency in a particular skill, for example, reading, may be high among one's age peers, but not particularly impressive among one's older peers. So, a great third grade reader may be reading at a level that's not that impressive for a sixth grader or for a ninth grader. So the point of this is just that a behavioral phenotype really should be thought of as emerging through the cumulative effects of factors influencing over the entire life of the individual. So why should we be interested in individuality? Why is that going to make a difference? Why is it important? Well, one reason that it's important is because really, some of the really seriously adverse outcomes that evolve do so disproportionately in people with certain kinds of phenotypes. So this is true of the risk for things like substance use disorders. It turns out it's much more common in people with certain phenotypes. Affective disorders, psychosis even, and things that we care about, like we've just been talking about adacademic failure, social dysfunction, even incarceration. So really, from conception to the onset of an adverse outcome or to a diagnosis, the evolving intermediate phenotypes are simply forms of individuality. People just start off kind of being maybe a little bit different in some very subtle way, and those people are somehow at a higher risk for developing these adverse outcomes. So we're still trying to understand why behavioral phenotypes actually diverge during development. And you might ask the question, well, is it related to genes? And I can tell you that there's kind of incontrovertible evidence that genetic variation does mediate behavioral phenotypes to some extent. But we really do not understand the processes by which genetic variation mediates a behavioral phenotype. Well, what about experience? Well, all of us as cognitive scientists, as behavioral scientists, recognize that the learning and experiences that people have are extraordinarily important in their developing behavioral phenotypes. But we still don't understand very well what kinds of experiences are most important and what critical interactions between experience and people's more constitutional factors, like genetic factors, might occur. And the most important question is, how much could learning and creativity and emotional resilience be enhanced if somehow we knew how to sort of optimally match people's experiences or the interventions that we offer them to their genotypes? So again, I want to remind everyone that, and this should be obvious, but we just think it's important to remind people that knowing things and becoming an individual take time. And we think this matters because it has implications for the time scale of the research. So this slide just illustrates that whether we're talking about very basic perceptual processes like the ability to detect coherent motion in a field of randomly moving dots, like you see, oops, is that going to work, up here? Or the amount of time that it takes you to cancel a primed motor response after a stop cue and a stop signal task, or these things that we think of as evolving through interactions with the culture, like reading and calculation, children exhibit a long period of gradual strengthening of these functions, suggesting that the mature functions depend upon mechanisms that act over a very long time period. But how do we understand these processes, and critically, how do we understand the variability? For example, why is it that this 14-year-old has literacy skills which are much more similar to most 8-year-olds? But on the other hand, this 10-year-old has literacy which is strong even among 16-year-olds. So this is kind of the model that we use to try to address these sort of basic questions about developmental processes leading to diverging behavioral phenotypes. We think of a sort of a behavioral phenotype like this, for example, expertise in mathematics or literacy, as developing as a function of, first, the hypothetical domain-relevant neural genotype. We don't know what that is yet, but we think there is one. And then sometimes environmental effects on the neural apparatus that's relevant to the skill or domain. For example, in some individuals, there's early brain damage or toxicity, or there may be epigenetic effects on the neural apparatus. But more generally, the cumulative experiences that the individual has encountering and manipulating material in the domain. And very importantly, there can be powerful explanatory interactions between these factors. So then we ask the question, well, how do we test and improve such a model? Because obviously, as you can tell already, it's going to be very complicated. And one approach that we think is important is to begin to accumulate large, informative, high-dimensional data sets. And we think they have to include a full assessment of genetic and, when possible, epigenetic variability among the individuals. We happen to think that it's important to have non-invasive imaging and other neurophysiological measures that are sensitive to differences in the neural architecture and also to the status of the brain's biological development, because as you'll see in a minute, biological development of the brain goes on for a very protracted period. And finally, we have to really add to this kind of information a much richer corpus of concurrent behavioral and experiential assessments so that we really can begin to model the effects of different kinds of experiences, different kinds of exposure, and other environmental factors. So I'm going to talk for a couple minutes now about a step that we tried to make in this direction with the PING project for the Pediatric Imaging, Neurocognition, and Genetics. This is a project that we created and hosted and coordinated here at UCSD. And I'm going to talk a little bit about its infrastructure, a little bit about the data that we collected, some about what we learned about brain development that we didn't already know from the PING cohort. And if I have time, and I may not, but I'll talk a little bit about, as an aside, about genetic parcellation of the cortex and some associations between anatomy and behavior. But let me talk a little bit about Ping. This was, we were lucky enough to get one of those large stimulus package grants about 5 years ago. And this is the title of the grant here. And what we did was to set up a multi-site study. I know you probably can't read all this, but we hosted the coordinating core here at UCSD. We partnered with Anders Dale and his multimodal imaging lab, who handled most of the imaging aspects of the study. We also partnered with people at Scripps Research Institute, who are our genomics core. But we had really a dream team of developmental cognitive neuroscientists and pediatric neuroimaging folks at all the data collection sites around the country. And there ended up being 10 of those. And we had really only one specific aim, which was to create a publicly available pediatric imaging genomics data repository, which did not exist at that time. There were a couple of similar ones in adults, mostly in aging, but nothing in children. And we studied kids 3 to 20, and we have now collected data on and have made available data on more than 1,400 kids, not every single phenotype, but at least something from each of those. And this was one of those very time-compressed stimulus package projects. It was supposed to be over in 2 years. Took us 4, but we did the best we could. But we could only get a limited set of measures, but we got some critical background information. We used this brief NIH cognition toolbox to get 8 primary cognitive measures from everybody in the cohort. We got some social and emotional phenotypes in a subset of this. And with Anders and Thomas Ernst, we were able to apply a pretty much state-of-the-art sort of high-throughput multimodality brain imaging protocol. And, of course, we got genome-wide genotyping. So I'm just going to show you a little bit of the data that are now, and the data have all been collected now, and they're all publicly available, and they're available through the Ping portal, and along with data exploration informatics that allow you to model the data. But I'm just going to show you some of the contents of the data. These are just the 8 basic cognitive measures that we collected, and it just shows you, I know you can't read this, but you can see that, each one of these behavioral measures has strong age dependence. This is just the increase in performance with age. But each one has a little bit different function, probably in part because how differences in the way these cognitive functions develop, but probably also because of some of the psychometric limitations of these brief tasks. And I'm just going to show you that these are basically measures of working memory, episodic memory, speed of processing, attention, 2 measures of executive function, and vocabulary and reading. So we have those kinds of measures for every participant in the study. And we also were able to find out some kind of interesting things about postnatal brain development. Of course, we've been studying this for quite a long time. But because we were able to apply sort of newer methods that Anders developed, partly he started developing these methods actually when he was still a student here in Kaisai. But now they have been developed to a much more sophisticated level. And so we are able to very clearly separate the effects on different aspects, the age effects on different aspects of the cortical architecture. And in particular, we've been able to separate effects on cortical surface area from cortical thickness. And this is important because in previous studies, these 2 factors had been conflated, and so there was kind of a misunderstanding about how the cortex developed. But with this large data set, we're able to show, and this is just like, this is a little map for you. I hope you can see that better than I can, actually. But anyway, these are maps that show you at every vertex, at every point on the cortical surface, an estimate of the rate of change in surface area. So in these 3-year-olds and 5-year-olds, what you're showing here is that at every point on the cortex, the cortical surface area is expanding in excess of 2% a year. But you can see that by 7, although there are some limited areas that still are showing that rapid level of surface expansion, there's some deceleration in the expansion in other parts of the brain. And by 9, you should be able to see that there are a few areas where it actually looks as though there may be a little bit of contraction, though most of the brain is still very modestly, the surface area is very modestly expanding. By 11, those two, the contraction and the expansion, are pretty much in balance, and not surprisingly, the total surface area of the cortex stops increasing overall at that point. And after that point, you can see that virtually all parts of the brain begin to show evidence of modest contraction. So there's this inverted U-shaped function in cortical surface area, rapid expansion of the cortical surface area, followed by gradual contraction of the cortical surface. And, but the phase of those U functions is different in different parts of the brain. In contrast, what we measure as apparent cortical thickness inexorably goes down across this whole age range. And in fact, cortical thinning is never more rapid than it is in the earliest stages, and then it sort of decelerates across the rest of the age range. But you can see that in these bright blue areas, there's actually thinning of the cortex in excess of 2% a year as well. Now, what about the underlying white matter? Well, we've known for a long time that, that the brain becomes whiter with age and over a fairly protracted period. And so a larger and larger percentage of the cerebrum is white matter, and this is presumably associated with this very protracted period during which there is ongoing proliferation and differentiation of oligodendrocytes, and they mature and they begin to wrap their fatty myelin sheaths around the axons and the brain fiber tracts. But more recently, we were able to use new methods with MRI, diffusion-weighted imaging, to actually measure, to compute indices of biological maturation of individual fiber tracts in the brain. And this is just another summary slide from Ping that illustrates what now has been observed on several occasions, and that is that some fiber tract, and I know you can't read this, but some fiber tracts, like the corpus callosum here, shows a fairly protracted increase in biological maturation according to this metric, but then kind of asymptotes out here about age 10 or 11, whereas some of the other projection tracts, particularly the thalamocortical and striatocortical projection tracts, show a somewhat more protracted developmental course, and some corticocortical fiber tracts, for example, this one called the cingulum, which connects the medial temporal lobe structures to prefrontal cortex, a sort of a frontolimbic fiber tract, shows a very protracted maturational course and doesn't clearly seem to be asymptoting even out here at 20. So I think the picture, hopefully the picture that's emerging here is a very highly dynamic process that's both anatomically and temporally very complex. So one question you might ask is if it's possible for us to integrate this information in some way across these many different phenotypes to get a deeper understanding of brain development as an integrated process. And so we tried to do this, the result was published in Current Biology, and it was the cover article. And so what we did is to apply a regularized nonlinear modeling and cross-validation method that was developed by Anders Dale that kind of rigorously corrected for overfitting. And we found that we were able to predict the age of individuals much more accurately than we expected, given the individual difference variability that we saw in the individual phenotypes. And this is using a set of 231 phenotypes. And I am going to let you, I'm going to let Tim Brown, who's the first author of this paper, talk to you, because he did this in a video abstract. And it's
First, we show that structural MRI scans of the brain can be used to predict a child's age with much greater accuracy than has been possible with other types of biological measures. In 885 participants from 3 to 20 years old, using a combination of brain measures from MRI scans, we predicted everyone's age within about a year on average, explaining more than 92% of the variance. What this means is that we've uncovered a developmental clock of sorts within the brain, a biological signature of maturation that accurately reflects age differences regardless of other kinds of differences that exist across people. This essentially means to measure where a person lies along a continuum of biological development made up of periods of different kinds of growth and changes over time, which you might call phases.
Okay, now my digression. I want to talk a little bit about the genetic architecture of the cortex. This is some work that we've done in collaboration with our larger imaging genomics group here at UCSD. Now, we've known now for a while that that functional areas of the mammalian cortex are established by gradients of gene expression, that is in transcription factors, and of secreted morphogens in the neocortical proliferative zone during the embryonic period. And this is just this is some work that's been done a lot of this work has been done by O'Leary here in La Jolla. And you can see that this is just kind of the pattern of regionalization that you see in the rodent brain. But what we were able to do was to take a large twin cohort from a study called VETSA, for Vietnam-era twin study, that's led by Bill Crimmen, and Anders Dale also is involved heavily in this project, and compute a large genetic correlation, which is the kind of thing you can do with twin studies, of the surface area expansion at every vertex on the cortex again, with every other vertex on the cortex. And in doing this, you can then apply, and this is an analysis that Chi-Hua Chen, who's the first author of this paper in Neuron and another one in Science, she applied a fuzzy cluster analysis to reduce this matrix, to analyze this matrix of genetic correlations. And this is the result of the 4-cluster solution. And what this means is that these vertices, the cortical area expansion and these blue vertices have higher genetic intercorrelations with each other and relatively lower genetic correlations with the vertices in the other areas, shown in different colors. And so you can see here that it's revealing something about the underlying genetic architecture of individual differences in sort of cortical shape. And you can see that there's a little bit of similarity to the pattern that you see over here in the rodent. Now, obviously, in the human brain, there's a lot more territory devoted to the frontal lobes than in the rodent brain, but I think the pattern is recognizable there. And it turns out that with this data set, the optimal cluster solution was for 12 clusters. And so here are the 12 clusters, sort of shown, the locations of them are shown in different colors. And this is not a functional parcellation of the cortex now. This is a parcellation of the cortex based on these underlying genetic covariation patterns, right? And so it's kind of a genetically informed cortical parcellation method. And we've used these genetic parcels to sort of do some studies looking at evidence for genetic mediation of cortical functions since then. And this is really interesting, but of course with twin studies you don't get any information about the actual genetic variants that give rise to these effects. But we were able to use the large ping cohort in collaboration with other consortia, with adult imaging genomics data sets, to discover and then replicate a particular variant that turned out to be in this GPCPD1 area, which had a strong association with the relative scaling of the occipital cortex. And this is just a vertex map showing the degree of association between the allele groups in this SNP in the GPCPD1 gene. And you can see the strong association between the relative size of the occipital cortex, or the areal expansion of the occipital cortex in this marker, and some sort of unexpected opposite direction associations in these areas shown in blue. And by the way, in the ping portal that we've now provided for this large imaging genomics data set, we've actually put into the portal an exploratory GWAS module so that people can actually go into the data set and do a GWAS of any variant that they're interested in that's available through the ping cohort. So now I'm going to talk a few minutes about the associations that we're seeing between behavioral measures that we collected in ping and neural phenotypes. And I can just say, to summarize, that there are associations even among children of the same age. And this is one of the first things that we published out of ping, which you'll remember that this is one of those distributions that you saw earlier. This is from a classic flanker task. Most of you guys from cognitive science probably recognize this classic flanker task, in which, in this case, kids have to press a right button or a left button based on whether the middle fishy, not arrow, but fishy, is pointing right or left. And as you probably can imagine, when the flanker fishies are pointing in the same direction as the central one, it's a little easier to do this correctly than if the flanker fishies are pointing in the opposite direction. This incongruency effect causes a reaction time cost. And this reaction time cost is related to the degree to which the participant exhibits sort of freedom from perceptual interference. And you can see that kids get a lot better at this. They show much less interference with age. And we can show that even when you control for age and gender and socioeconomic factors and even genetic ancestry, the relative expansion of this region in the anterior cingulate cortex is associated with greater freedom from perceptual interference in kids of the same age. And this region has been strongly implicated in this kind of function in many other contexts in adults and even in other groups of children. And this is just another example where we were interested in one of these genetic parcels that I showed you earlier. This one happens to delimit the ventromedial prefrontal cortex very well, the one shown here in pink, which again has been implicated very strongly in anxiety and depression in many other contexts. But we were interested in whether it was associated with self-reported high levels of anxiety in children in the pink cohort. And so what we found was indeed it is, that the cortical surface area in this region is lower in children who report high anxiety, but more importantly to us, really this is attributable to the fact that there's some evidence that children with high anxiety show a later expansion of this region across the age range. Although these are cross-sectional data, that's sort of the pattern that we observe, this interaction between age and anxiety. And just before I stop talking about these, sort of giving you these highlights, I want to mention that we have a new learning research hub, which is funded by the National Institute of Child Health and Human Development. And we call it the Role of Emotion and Choice Learning Research Hub. This is to create new methods for assessing both the developing math-relevant conceptual skills and developing math domain affect in young children just as they're entering formal education, just as they're making the transition from kindergarten into first grade and second grade. And then to use these methods, hopefully for assessing both their conceptual skills and their beginning affective responses to improve our models of the roles of brain development, neural architectures, conceptual factors, and the reward dynamics that are going with the learning that they're doing in accounting for languishing calculation abilities in children who we know from other sources are at high risk for math failure. And this is just showing you our very beginning. You can see that we're studying these children longitudinally, so we're looking to see how they're skilled. This happens to be calculation, a measure of calculation, and these are our PING participants who are older. But you can see that these little spaghetti plots are to show that we're following them over time and following their individual trajectories of learning so that we can link them to these different biological, conceptual, and emotional factors. And this is just a very preliminary look again, suggesting that, again, we're going to see some association between the cortical phenotypes and these calculation skills. That suggests that, that children with stronger calculation skills for age seem to have somewhat areal expansion in this middle temporal area in the left hemisphere, and in this inferior frontal region in the right hemisphere. This has been associated with cognitive control. So brain behavioral phenotypes are related to individual differences in brain architectures. And in general, more mature cognitive and social emotional phenotypes correlate modestly with more mature neural architectural characteristics. The associations remain after controlling for age and global parameters. It appears not that there are some kids who are just advanced in their development and who have better functions, but that the profiles of their kind of strengths and weaknesses are mirrored in patterns in the neural architecture. And recent evidence suggests that some behavioral phenotypes may be associated with developmental endophenotypes. That is, the course of development itself seems to map on with the outcomes, onto the outcomes. And at these, we also have some evidence I didn't have a chance to talk about that some of this is genetically mediated. Okay, so here's our conceptual model for sort of how we would translate this kind of information into better interventions. So it seems possible to us that variations in early genetic patterning in the brain, for example, of the cortex and of the connecting fiber tracts, and probably many other parameters that we don't measure with these measures that are not strictly architectural, may introduce very subtle biases in the infant's sensory motor processing. And in addition, other developmental phenotypes may continue to emerge throughout childhood, reflecting changes in gene expression patterns that we know exist, and these may influence the ongoing maturational processes. And in part because of these effects, as development proceeds, some types of information may garner more or less salience to particular individuals. So, in some individuals, this may lead to relative inattention to and languishing development in some domains relative to others, for example, reading or calculation or social interaction. And these differences may be accompanied by critical effects on the reward dynamics, what we generally think of more in terms of whether someone is engaged by a particular domain or avoids a particular domain. And these effects may further enhance or suppress a developing behavioral phenotype. These conceptual and reward effects are likely to influence both the behavior of the child and the associated neural circuit development through neuroplastic means. And the implication is, we think, that if subtle neural effects that are present early in life cascade into individual differences through the development of perceptual motor and motivational biases, then it may be more useful to think of these experiential mediators as the proximal causes, rather than the neural or genetic origins per se. So, importantly, domain biases and reward dynamics that constrain learning or that in some way limit functional adaptation are likely to be preventable and reversible with intelligent, and by this I mean adaptive interventions, like some of the ones we just heard about. But critically, this intelligence depends upon a much better understanding than we have now about the ways that genetic variation and experience interact to make us unique individuals. And so I'm going to stop there and acknowledge my collaborators. I want to acknowledge all the investigators in PING and REACH and the Center for Human Development, of course, the funding agencies that are funding this research. You heard from Tim Brown. Eric Newman did the anxiety work. Natasha Akshamov is a partner on all of this for the assessment aspects and has her own program on preterm birth. Wes Thompson is a biostatistician extraordinaire. I couldn't do any of this without my buddy, without whose good offices, you know, I would be helpless. Anders Dale, one of your fellow alumnus from the Department of Cognitive Science. So I'll stop there.
Okay, well, I know everyone wants to get coffee, so I think we'll continue our tradition of one question for Dr. Green.
I saw a hand over there somewhere.
You can handle it.
Dark back there.
Could you put back up your slide of the anxiety? That's a really weird way for me to describe it, but I think you know what I'm talking about.
There we go.
Okay, so I'm sorry. This is going to be a series of slightly dumb questions, not one big smart question. Are these two? We can sort of imagine this as two people, right? There's low anxiety and high anxiety, and when they're 10 years old, there's differences in their brain. What's going on when they're 22? Are their brains kind of the same? And what's happening with their behaviors?
Right. Let me say again that this is a cross-sectional study.
Well, okay.
And so, because I know it kind of sounds like when I talk about it that it isn't, and that's misleading. But it's this large cross-sectional study. So what we're really reporting here is an interaction between anxiety and this ventromedial prefrontal cortex size, right? So, and age on that size, on age and age on that size. And actually we've done this model both ways, and that's one reason why it's a little confusing. But really what it means is that anxiety is strongly, actually, you know, I actually have a slide. Let me just, I think I have a slide that actually shows that better. See if I can find it.
I guess my question is, it looks like they're converging when they're 20,
Right.
and they're different when they're 10, and I'm trying to understand whether there are still behavioral differences, even though you might get two people who have very similar looking brains when they're 20 on the
Right.
factors that matter. Their
Right.
behaviors are still, whether or not they're a high anxiety person is still divergent, and that seems to have more to do with what their brains looked like when they were 10
Right.
than when they're 20, and I can't tell whether I have that
So
right.
that's definitely one of the possibilities that we think is consistent with this pattern. It's not the only one, but that's what we think is consistent with it. And you can see here that's kind of what we're showing here. I don't know if this is working here, but this is showing the association for every vertex sort of cortical surface area with anxiety at 7, at 14, and at 21. And you can see it kind of goes away. This is true for cortical thickness. I didn't talk about this, but it's also true for cortical thickness, but it's a more global effect in cortical thickness. So it really says the association's here, it's strong here, it's still present, weakening just a little bit at 14, and it's gone at 21. So that is consistent with the idea that it was somehow the way that the system developed that was different. But we can't be sure. Well, all we know is that in the 21-year-olds, we don't see an association. So we need to do that development. We need to do that longitudinal study to be sure about what's going on here. But that's one of the hypotheses that we have. That's one of the interpretations that we have as well. It could be that individuals here because we do have individuals at 21 who report higher anxiety, but we're talking about present anxiety. That's the other thing. So we can't say for sure what the anxiety level was earlier, right? So we're just talking about current. So we know that these kinds of temperamental phenotypes are fairly stable. They're fairly stable over the age range. Not entirely, but they are fairly stable. So it's a good question. I wish I could answer more definitively, but I think you got the pattern right, though.
Thank you.
This is what you get when you ask a PhD.